Welcome to the Allen School colloquium. So today we are going to hear about some of the research we do in the AIMS lab. AIMS stands for Artificial Intelligence for Biomedical Sciences. And then our recent research focuses on a broad spectrum of problems in machine learning, developing explainable AI techniques, also known as interpretable machine learning, and identifying cause and treatment of challenging diseases such as cancer and Alzheimer's, and developing and auditing clinical AI models. We just briefly introducing who we are. We are a group of passionate researchers and brilliant researchers. I'm not biased. Consisting of a PhD student in the Allen School and then MD PhD student pursuing PhD in computer science, who are interested in research in this intersection, exciting intersections of machine learning, biology, and medicine. So here for some of the people who are exploring some other intersections are indicated with arrows that measure the steepest gradient direction. Okay, so so today we are going to hear from 4 speakers in this diverse set of areas about some of their work. And during the talk, you are welcome to ask clarifying questions. Either raise your hand, those who are here, or type on the chat channel on Zoom. I will constantly monitor the chat. The first speaker is Joe and Alex. They are MD PhD students pursuing a PhD in computer science.
Thank you for the introduction, Sue-Ann, and thank you all for having us. I'm Alex.
And I'm Joe.
So for our portion of the talk today, for about the next 10 minutes, we'll be telling you about some work that we did last year around the height of the pandemic. And this work basically centers around auditing some machine learning models that aim to detect COVID-19 in chest x-rays. So I'll start off with a brief overview of our talk just to help you stay oriented. Overall, we hope to demonstrate to you that there's a number of published artificial intelligence models that detect COVID-19 in chest x-rays, but they are not robust. And the reason they're not robust is because they take shortcuts instead of looking for genuine biological evidence of disease. So we'll start by going through some brief background on the topic. Then we'll look into how robust these models are. We'll follow that up by digging deeper and trying to explain why exactly these models fail. Based on that information, we'll look on how we can improve these models. And finally, we'll finish up with a few brief conclusions. So again, we'll start off with some background.
All right, and so the inspiration for this project kind of started at the beginning of the pandemic last spring, when we began to notice that there were a ton of these different deep learning models to try to detect COVID-19 from chest x-rays. And in the intervening time since then, by late 2021, it's established now that these sort of models are now being deployed in clinics kind of across the world. And one potential problem that Alex and I noticed when reading papers about these models was that many of them had really suspiciously high reported performance, kind of beyond the bounds of what we might have expected would be reasonable for a human or a deep learning model to kind of attain.
So trying to figure out why this performance might be so suspiciously high, we noticed that there is a commonality between a lot of these published models, and that was in how they obtained their training data. In all of the cases that we looked at, they would obtain their COVID-19 negative chest x-rays from one source, for example, one hospital, and they would obtain their COVID-19 positive chest x-rays from another source, like another set of hospitals, or even by extracting images from online journal publications.
And in case it isn't immediately apparent to you what the problem might be there, the issue is that kind of any systematic differences between these data sets could be used as a shortcut to determine COVID-19 status. So not just medically relevant pathology, but any sort of system level differences between these kind of data sources. And so the hypothesis that we wanted to test was that these really high-performing published models rely on shortcuts to a similar or even maybe a greater extent than they relied on actual genuine medical pathology to predict COVID-19 status.
So now we'll go into our tests and to how robust these published models are. And specifically, we wanted to test these models on data from new environments with the assumption that if these models depended on shortcuts, those shortcuts might not generalize to new environments very well. To perform these tests, we needed 2 data sets. For the first data set, which we call data set 1, we used the data that was originally used to train these models. And again, this consisted of COVID-19 negative chest x-rays from one source and COVID-19 positive chest x-rays from another source. We then built a second data set in a similar manner to the first data set, again taking COVID-19 negative and COVID-19 positive x-rays from different sources. But we were careful to ensure that all of the x-rays in data set 2 were completely external to those on data set 1, such that any shortcuts that might be present in data set 1 may or may not be present in data set 2.
And so once we had created these 2 data sets, what we did is we would take a model, train it on the original same data set 1 that these models that had been published had been trained on, and then evaluate it both on held out test data from this same data set 1 and held out test data from data set 2, which is like a completely different data set. And so when we evaluate the predictive performance of these models, trained on data set 1, they perform really, really well on held out images that weren't used for training from the same data set 1, but really, really poorly on images from data set 2. And the performance here you can see drops by about 50%, consistent with these models relying more on shortcuts than on real, true generalizable signal. And we see the same thing when we do the reverse procedure and just train first on data set 2 and then test on held out images from data set 2 as well as images from data set 1. And so again, this large generalization gap is consistent with sort of a shortcut learning kind of picture.
So the results we showed you so far are just from a single primary architecture that we looked at, but you might also wonder if this holds true for other architectures. For example, there is a variety of architectures that were specifically designed for detecting COVID-19 in chest x-rays, and there are just a lot of different architectures out there. So we went ahead and tested them, and in all cases, we found that they did not have very good generalization performance.
Right, and so now that we've kind of shown that these models aren't really particularly robust and don't generalize well outside of the development set that they kind of had been trained in, we wanted to try to build an understanding of what shortcuts these models were relying on and why they failed to generalize more broadly. And to try to answer this question of what was important for these models' predictions, we used a variety of methods from the field of explainable AI, including saliency maps, which try to highlight important regions of an image for a model's prediction, generative adversarial networks, which allow you to kind of generate counterfactual images. What would this image look like if it had COVID-19 or if it didn't have COVID-19? And then finally, just manually manipulating images by moving around text markers on them or other things like that and seeing how that impacted the model's output.
Based on those explainable AI techniques, we were able to identify a number of specific factors that these models relied upon to make their prediction of a patient's COVID status. Some of these things were appropriate and some of these things were inappropriate, and that the models should not be relying on these factors. So among the good things that the models were doing is that they would often look at the opacity of the lungs or how light or dark the lungs are. This is something that a doctor might also look at in determining whether a patient has COVID-19. However, there are a number of inappropriate things that the models examined. For example, the models often looked at specific textural markers. They would look at the position of the patient's shoulders in relation to the top of the image. They would look at the appearance of the lateral image edges. They would examine the patient's diaphragm, which is the muscle you use for breathing, but not something that's implicated in COVID-19. They would examine the patient's sex. And they would also examine something called the radiographic projection. So in all of these cases, these factors are either demographic factors that a physician would already have accounted for, or they're artifacts of the image acquisition process. And the way that an image is acquired might differ between different hospitals or different departments that have differing rates of COVID-19. So for example, in the case of radiographic projection, this has to do with the way that the x-rays enter a patient. So they can either enter the patient through their front and exit through their back, or enter through their back and exit through their front. And depending on which scenario you have, you end up with an image that looks slightly different, and that can end up correlating with COVID-19 status.
Right, so now that we had a little bit of a better understanding of kind of these hospital or data set specific factors that the models were relying on, we wanted to try to figure out how we could improve these models and make them generalize more broadly. And the thing that we tried is basically, in dataset 2, we had this scenario where positive and negative images were collected from different hospitals. We then, using some newly published data, were able to assemble a dataset 3 where both the negative and positive images were taken from the same set of hospital over the same time period, so there's less possibility for source-specific confounding.
So we trained models on both dataset 2 and dataset 3, and then we tested them both on the same external data from dataset 1. And what we found is that dataset 3 had significantly higher generalization performance. In other words, it's important to train models on data that has less potential, that's less conducive to shortcut learning.
Yeah, and so just to quickly wrap all of that up, in conclusion, we found that these highly cited COVID-19 detection models relied more on shortcuts often than relevant pathology, and showed that testing on held out data from the same hospital system or sort of source data set that you used, even if you didn't use it for training, is often inadequate. We then showed how explainable AI could reveal shortcut learning. And I think all of this auditing work, especially in light of like the sort of naive unreliability of some of these base models, highlighted how model availability is really important for trust and safety. So yeah, thank you.
Might it not be more effective to train on all the data sets so that it can also take into account those differences? Because you said that you were training now on a third data set, both the positive and negatives. Wouldn't it then be a little more confused if it's looking at a second data set for the testing? If you know what I mean.
Yeah, so I think there's generally this idea in the machine learning world that if you train on just getting more data in general, that oftentimes these differences end up kind of washing out in the data and you can learn a better model overall. In this case, we didn't specifically look at models that were trained on both dataset 2 and dataset 3 simultaneously. That could be an interesting thing to look at. But definitely in this case, we're finding that making sure that the shortcuts aren't present to learn in the first place is important for building models that don't depend on those shortcuts.
Yeah, I think it's also a thing where it's possible that since we had a relatively small number of data sources on the order of like 1 to 2 per data set, maybe some of this washes out if you have on a much larger order of number of sources, like dozens or hundreds, but it seems like it mostly gets worse when you kind of add in additional sources at this small number of sources level.
Wow, there are many questions, but there was a question on the chat during the talk, and then let's quickly. I think it's a quick question. So there's this Kanan.
Oh yes, this is Kanan, and it's a very good presentation. Thank you. I really appreciate that. I had a question regarding the conclusion part. How you were able to conclude that the models that you have used behaves badly because of the effects that you are showing it as a shortcuts, having one set of data from one system and another set of data from another system.
Sorry, I think I had a little bit of trouble hearing the question. Did you pick it out exactly?
I only got bits of it, yeah.
So how do you conclude that the models that you have used worked or predicted badly with dataset 2? My question was, if you have trained them, if you have developed the model using dataset 2 and validated dataset 1, did it behave well?
So I think you're asking if we train on dataset 2 and test on dataset 1 if the performance is good. And when we looked, it's not, no. So with dataset 2, there's still source-specific confounding that's happening. I think the image sources are somewhat more similar than in dataset 1, but they're not perfectly matched like dataset 3. So the generalization performance is still quite poor.
Okay, thank you.
Okay, just one very quick question.
Do you think it would make sense to either perform some sort of data augmentation or some dimensionality reduction that takes out some known shortcut factors? Like, would that be something that makes sense?
Yeah, and I think that's something we've actually done in another research paper we published before this one, where not for COVID-19 detection, but for pneumonia detection, we were able to kind of remove the model's dependence on that AP-PA projection and found that we were able to improve generalization performance. In this case, because the hospital source information and COVID-19 pneumonia information are so tightly correlated, you basically have a problem where you don't have a lot of like overlapping support between source information in the image and COVID-19 information in the image. So I think it's really, really hard in this case.
Oh, okay.
All right, well, thank you all.
Yeah, thank you.
Okay, thank you.
Hi, everyone. I'm Ian, and my talk today is about all of explainable AI. And that probably sounds like a little too much to cover in such a short talk. And yeah, in reality, we're not going to talk about all of explainable AI. But the idea is I wanted to tell you about some of the core ideas that most explainable AI research is built on. Because it's a relatively new, cool, exciting field, but it's a bit fragmented right now. And these ideas that we've come up with, we think they provide a pretty helpful perspective for understanding a lot of the work that's been done. So that's what I'm going to tell you about. Here's a really short outline. First, I'm going to tell you what explainable AI is about. And then the main thing is I'm going to show you a recipe, kind of a shared recipe for making most explainable AI methods. Okay. So what is explainable AI? Well, ever since machine learning has started working, everybody wants to use it, including people working in some high-stakes domains like medicine, law, and finance. And these are domains where we obviously want our models to be as accurate as possible, but we also care about understanding how they work. We'd like to understand how the models think or what information they leverage so we can see if they think like doctors, or if they've discovered something new, or if they're doing something they shouldn't, like relying on spurious signals. And the problem is that the models we use these days, they're not designed for transparency. They're designed for flexibility and performance, and that's great, but they're black boxes. So we can't just look at one of these models and learn anything about how it's thinking. So that's where explainable AI comes in. Explainable AI is this field where people try to work with whatever black box model you give them and create algorithms that can give some degree of information about how the model's working. All right, so if machine learning systems are making medical diagnoses, hopefully we can say why and identify the information that's driving their predictions. Or for a consumer finance example, if someone applies for the Apple Card and the machine learning system denies them, hopefully we can figure out what it is about them that led to that decision and maybe what they can change to get approved the next time. So like I said, it's a big field. There are a lot of papers, many ways to approach a problem this open of saying something about how a model is thinking. So the key for us today to cover most of explainable AI is a shared recipe, a recipe that describes most explainable AI methods. So let's start with some intuition. Let's say, hypothetically, you're coughing a little bit, You go to the doctor takes an x-ray, and the doctor says, bad news, you've got COVID. You might say, are you sure? How do you know? This hypothetical doctor is very inconvenient. They don't like to explain their decisions. They just make diagnoses. So that makes it a little bit more difficult to understand how they're making diagnoses, but there is a way we can learn something. And the idea is really simple. The idea is we can take the scan that they took, block out certain image regions, and basically present that new image to the doctor and ask them to redo the diagnosis. And by seeing how the diagnosis changes, maybe we can learn something about the role that that information played in their diagnosis. So for example, if we block out this region, the doctor might say, still looks like COVID to me, sorry bud. If we block out this region, they might say, still looks like COVID. But if we block out this one and they say, all clear, no signs of COVID, that tells us that the information that was driving their diagnosis was right there. And it turns out that that's what most explainable AI methods are doing. They're doing the same thing, but they're doing it with machine learning models. So with that intuition in hand, let's talk about the recipe. It's a really simple recipe. It takes 3 ingredients. It's like choosing a meat, a vegetable, and a spice. And with these 3 ingredients, you have an explainable AI method. So the first ingredient is how to remove features from your model. And what that means is you want to withhold some information to see how the prediction changes. But you can't just not give the model the features, because the models we typically use actually require all of the features. You need to give some value. So you need some trick, basically, to remove features. And for example, one really simple thing you could do is you could say, all right, I'm not going to give the model these features, so for them, I'm going to set them to their mean. That's a neutral value. I'm just going to set them to their mean, see what happens. Alternatively, you could sample some new values for those features instead of setting them to the mean. There are other things you can do. Okay, the next ingredient is you need to choose the quantity that you're going to analyze. And what that means is, as you're removing different groups of features and seeing how the prediction changes, you just need to choose really specifically, are you looking at how the prediction changes or how the loss changes, or maybe even how the data set loss changes if you're removing those same features for multiple examples in the data set. So that's the second thing. And the last ingredient is you need to choose how to summarize each feature's role. And what that means is, so we're going in, we have this model, we're removing different groups of features, seeing how the predictions change. But there's a lot of different subsets of features we can try, an exponential number of subsets. So that's way too much information to communicate to a user. We're not going to do that. We need something a little simpler, something more concise. And what we typically do is we try to associate one number, a score, with each feature, usually called an attribution score. And that number for that feature represents that feature's impact on the model. So just to give you a really simple example, one way that you could do that is you could say, here's what the prediction is when I have all the features. And if I remove just one feature, here's how the prediction changes. And if you look at that delta, the delta between those two predictions, that number could be your attribution score for that feature, what impact it has on the model. So that's the recipe. You just have to choose these 3 things, and you can explain each feature's role for any machine learning model. And for any combination of these choices, for these 3 things, you get a different explainable AI method. And we were a bit surprised when we figured this out, when we started looking at things this way, but it turns out this is what a lot of explainable AI research has been doing for the last 10 years, experimenting with different combinations of these things and introducing new ways to do one of these things or another one of these things. But they're all kind of following this recipe. So when we started seeing things this way, we wrote a paper about it, and we figured out there are actually 26 published papers that all follow the recipe. In this table, each row is a different paper, and each column represents the choice that they're making for that part of the recipe. And these aren't just any old 26 papers we found. These are some of the most highly cited, widely used tools that have been developed in the last 10 years, and they're all following the recipe. And a couple of these, by the way, were developed here at UW, Lime and Shap. You may have heard of those. So really, it's more than a recipe. It's a framework that lets us unify all this work that's been done, understand it in a really concise way, and better reason about what the differences are and what the advantages of different approaches are. So to give you an example of one of these methods, SHAP, this is a method that was developed in the lab a couple years ago by Scott Lundberg. And the paper is a bit of a technical read, but you don't have to read it. We're going to make it really simple right now. Here's what SHAP does. SHAP wants to remove different features from the model, right? And when it says, I'm going to remove these features from the model, it samples some different values for those features. Then, when it's removing different groups of features, it just looks at how the classification probability changes. The last thing is, to calculate a score for each feature, it calculates this thing called the Shapley values. It's an idea from game theory, and to put it simply, it's like you want to understand what difference it makes when you introduce this feature, but the difference that a feature makes kind of depends on which features are already there. So Shapley values are basically a smart way of considering all the groups to which you could be introducing this feature. But I'm going to spare you the equation. So to give you a taste of what you can do with tools like this, here are a couple examples where we're trying to understand a model's global overall behavior. So on the right, we're explaining a model to try to understand the key mortality predictors. So you see like age, gender, blood pressure, those are big predictors. On the bottom, we are analyzing a different model to try to understand which genes carry the most information about someone's breast cancer status. And here's an example where we are explaining individual predictions for a computer vision model. And the explainable AI method here is identifying the key regions that were driving the predictions for these images with the different objects and different dog breeds. And the same technique is, of course, applicable with different kinds of images, like biomedical images. So anyway, this is our paper, Explaining by Removing a Unified Framework for Model Explanation. Like I said, the paper kind of starts by figuring out that there are 26 papers that follow this recipe. And then after that, we consider this kind of interesting question. I think it's pretty interesting. And the question is, why is it that so many researchers were basically doing the same things without even exactly realizing it? In other words, what is it about this approach that's so natural, so intuitive to so many people that didn't even put it down on paper that they were following this recipe? So to flesh out the merits of this approach, figure out what really makes so much sense about it, we found some new links between explainable AI and game theory, psychology, and information theory. So to wrap things up, I'm going to just do a really quick summary. The first thing is there are some recent papers that say if you're doing explainable AI, maybe you should read more social science research to try to understand a little bit better how humans understand complicated things and build on that. It's a good idea. So I did read some social science research, and it turns out this basic idea of removing features to see what impact it has on the model, there's definitely precedent for that in psychology research. It's a really natural way to try to understand something. So we think this actually is linked with human psychology. The next thing is we uncovered some new connections with game theory. So for methods like SHAP that use Shapley values, one of the big selling points was that it has this basis in game theory. But it turns out that if you look at things the right way, all 26 methods in that table, every method that follows the recipe can be understood as building on ideas from game theory. So they're all game theoretic. That doesn't mean they're all as good as SHAP. And the last thing is we derive some new connections with information theory. If you don't know what information theory is, that's okay. But the idea is previously, only a few of those methods in the table claimed some connection with information theory, this subfield of math. And the idea was that these methods were more theoretically grounded than the others. They weren't just some heuristics. But it turns out, once again, all 26 methods, if you tweak them the right way, they're all related to information theory. So they're all pretty theoretically grounded and not just heuristics. So yeah, that's an overview of the paper for you. You now know the basics of explainable AI. And if you want to learn more about the field or the tools that are out there, I think our paper makes a nice starting point. So thanks.
I don't see any questions on the chat. Let's see if there is a question here.
Just like on a very high level, I was wondering, why do you think is the reason that people haven't been like studying the XAI from the perspective of like knowledge formation? Like for example, going back to your example of the doctor diagnosing the, from a chest X-ray, like is like instead of removing certain features, why isn't it like impossible to kind of know how the doctor formed their knowledge and then like go from the other direction?
Oh, well, I guess I don't know if I'm understanding this exactly correctly, but I should maybe clarify. The doctor thing is just a hypothetical. It's the doctor in that case is kind of like a model in the sense that they're making predictions but not explaining anything. So for us, like understanding how the doctor came to gather their knowledge is kind of analogous to how the model was trained. And I guess some people have tried to like trace the training algorithm and learn something from there, but other people would probably say there are many ways to arrive at this model, and the particular way that this model was trained to arrive there doesn't necessarily matter to me. So does that answer it at all?
Okay.
Well, I guess maybe a related question or a different way to approach it is it seems like the recipe you've really outlined very elegantly here can map you from ranges of inputs to outputs, but it doesn't necessarily lead to a concise explanation, right? You know, I could think of, you know, an exhaustive exponential sized table versus a very compact decision tree. Mm-hmm. This recipe doesn't necessarily tell me how to go to the compact explanation. And
yeah, it does not give you like a very small decision tree that's like representative of how the model works. That's right.
Yeah.
Yeah.
And so is that sort of an unexplored frontier of explainable AI? Would you say that is sort of definitionally not explainable AI? Is that not important in the field? Like I think this is, I think when I heard knowledge formation, I started thinking about like, there's something in that decision tree that's getting to the actual knowledge, as opposed to the mapping.
Yeah, so yeah, like you're saying, the format of these explanations that all of these methods generate, it's always, you have your model, you had your input that you put into the model, it made a prediction, and the way that I'm going to explain it is by saying what each of the features' role is, by associating a score with it. And that's actually, yeah, it's not as concise as giving a small decision tree that is representative of how the model works. There are some papers out there that try to find something like a small proxy model that is similar to the original model, but it's sort of tough because the whole point of using a complicated model is that it works with patterns that a small decision tree can't work with. And so I should also say, because I'm trying to cover all of explainable AI here, there is kind of a tension in the field between people who say, you know, if you're working in a high-stakes application, you shouldn't even use a neural network. There's some people who think that you should use a simpler model that's like a decision tree in the first place, where you can just look at it and understand things. And that's a totally valid perspective. I think in the image context, you probably can't find a simple model that does a good job. So that's kind of where we've been working a lot, this place where we'll take any model you give us, any complicated model, and try to say something. But yeah, people are thinking about all these things. Thanks, guys.
All right, I'm Joe again, and I'm gonna, I think, try to present this pretty quickly because but back to talk again about a different project we worked on in lab about some of the particular difficulties of applying the sort of explainable AI methods that Ian just talked about in the field of cancer precision medicine. And to give a bit of background on the problem in general, The cutting edge in the treatments of cancers like the blood cancer acute myeloid leukemia, or AML, has increasingly become this combination therapy, where the rationale of combination therapy is that by choosing drugs that target complementary pathways, you can achieve greater anti-cancer drug efficacy with fewer side effects. But the issue here is that choosing optimal combinations of drugs is just a really hard problem, right? There are already hundreds of individual drugs that have been FDA approved to treat cancer, which means that there can be on the order of tens of thousands of pairs of these drugs, and even more if you considered drugs still in development or larger numbers of combinations than just pairwise combinations. Finally, this is all complicated even further by the fact that different patients, even patients who share the same cancer type like AML, may all respond differently to the exact same treatment based on the particular genomic characteristics of that patient's tumor. And so if you're a machine learning person, your first thought might be that this sounds like the perfect opportunity for kind of predictive modeling, right? Because you could collect a data set with a bunch of samples, including the characteristics of different patients' tumors, like their gene expression levels, and the characteristics of different combinations of drugs, like the structure of those drugs or a list of their biological targets, and use this data to train a machine learning model that hopefully generalizes well so that it can be used to predict how synergistic different combinations of drugs that you had never even tested would be for different patients. And so we tried applying this to this large publicly available data set of over 12,000 samples from many different two-drug combinations tested in hundreds of patients. And what we found, this figure's a little complicated, but the kind of takeaway is that complicated black box models like XGBoost, which is like a library that implements a class of algorithms called gradient boosted machines, outperforms simple linear models, which leads you to a kind of problem, right? A first problem here, which is that we know from the past work that like Alex and I just presented, that in high stakes scenarios, it's important to be able to interpret models to know that they're doing something that you can trust and making good decisions that'll work out in deployment. And in some sense, interpretability might be even more important in the context of pharmaceutical development, where discovering the particular biological mechanisms underlying drug synergy for different combinations of drugs can motivate future research into like new and effective drugs. But in trying to apply one of these feature attribution methods called SHAP, like the methods that Ian just discussed, to the high-dimensional, highly correlated gene expression data sets that characterize different patients' tumors, we ran into a really interesting problem. And so when we trained different XGBoost models on the exact same data set with different random seeds, or similarly neural networks with different random initializations, we found that these models that would get around the same test error on the same sort of data set would rank features very differently. So each row in this heat map is a model trained on the same data set, while each column represents different features, and the intensity of each cell encodes the importance of that feature for that model. And so if you have a background in stats or ML, this phenomenon might sound similar as something that Leo Breiman described as the Rashomon effect, or a multiplicity of good models with sort of similar test error that use really different explanations from the same data to make their predictions. And so this leads to a really natural question that if you're trying to build an ML model that you're going to have, it's like, how do I choose the best model, and which model's explanations do I really want to look at? And I think one of the most natural ways that you might think to do this is just look at the test error. And what we did here is we generated synthetic labels for this AML data so that we still had all of the AML gene expression features, but generated synthetic data so that we could measure how well the attributions lined up with the true ground truth. And what we saw here is that the y-axis here measures how well models' feature rankings align with the true feature rankings, while the x-axis shows you the model's test error. And what you see is something kind of concerning. While overall there is a large correlation between test error and feature attribution quality, within a particular model class, there's not a significant correlation, right? So maybe looking at test error helps you pick that I want to look at gradient boosted machines rather than linear models, but it doesn't help you understand like which of these XGBoost models do I really want to trust the explanations from. So that's kind of a hard problem. And so we tried a really simple but I think good fix that ended up working pretty well for us, which is just to try ensembling attributions over many of these well-performing but different explanation models. And so again, the y-axis here shows you feature attribution quality for individual models for linear models on the left and gradient boosted machines on the right. And the red shows you now, after you ensemble many of these models, the feature attribution quality increases. And so these ensembles of complex gradient boosted machines were then what we would apply in our final analysis. So once we had the more stable ensembled attributions, we tried auditing some sanity checks on our model and found a bunch of really nice things, like if you look at what genes were important for the drug venetoclax, a small molecule BCL2 inhibitor, you see a bunch of genes, including both its actual target, BCL2, and a bunch of other genes that are related to BCL2 in function. So that's really good. We also wanted to look to see what genes tended to be important over many different combinations of drugs to try to find general patterns driving drug synergy in AML. And we found two genes, MICE1 and DLL3, both of which had previously been linked to AML prognosis, that were both related to hematopoietic differentiation that were very important for our model. And the importance of this, both of these are linked to a cancer's stemness, or how similar it is to a stem cell, like, or how undifferentiated the cancer is. And this is further confirmed when you just check for enrichments at the top of this list of attributions for certain biological pathways. So yeah, I think we're definitely running a little slow. So we find that this is like an important trend across many different combinations of drugs. And think that these results, I think, are show the importance of really, being careful and thoughtful when trying to apply some of these feature attribution methods to some of these complicated, high-dimensional, highly correlated biological data sets. And this paper, if you're interested in reading more about it, is out now on bioRxiv. Thank you.
Question.
You mentioned at the beginning that the test performance was different, even though the feature attribution was different for various seeds. Were also the predictions the same, or just the test error?
Yeah, there was a really high correlation between the predictions from model to model. I didn't look to see what the exact number of discordant predictions from these models was, but yeah, there's like a very, very high actual concordance between the predictions from model to model.
I have one question. Genes are actually not independent, they are kind of structured. So the different attribution of the genes might due to the gene structure, they are co-expression. So when some gene highly expression, other gene will high expression too. So gene module might somehow to solve this problem, to embed this gene expression to a low dimension space to do this. Actually, it's quite like the ensemble model, I think.
Yeah, that's a great point, and that's something that we're looking at further now. I think the variation between good models, I think, still would persist at that level because we did generate some kind of higher level attributions at the level of enrichment of these attributions for certain pathways, and those did end up still, the sort of top pathway enrichments would vary from model to model still, but certainly less than the gene attributions did. So I think that's definitely a great point and starts getting things, thinking about, pathway representations of gene expression is a great direction that partially but doesn't totally solve the problem.
Sarah, you had a question?
I'll make it quick. I'm wondering if the ensemble approach will also help you close the gap between different attribution methods. So for instance, if you take into account uncertainty and your feature importance, that could help you explain why you get different explanations by different approaches.
Yeah, that's a really great point, and I think something that's definitely going to be interesting to look at more. That's awesome.
Maybe in the revision. Okay, so let's turn to the last speaker for today. Yeah, thank you.
Cool, thank you. All right, hello everyone. I'm Ethan, and for the last talk of the day, I'll be talking about contrastive latent variable models for biological discovery. So oftentimes when biologists or computational biologists are analyzing and exploring a new data set they've collected, they want to specifically explore variations that are enriched in some target data set as compared to a related background data set. So for example, if we collected a set of gene expression measurements from patients with some kind of cancer, we might want to explore variations in this target data set from the cancer patients as compared to variations that would also be present in some background data set of measurements from healthy patients. And we care about these like cancer-specific variations because they might provide insights into, let's say, different molecular subtypes of cancer that behave differently, and might give us insights as to why, as is the case with AML, as Joe described, different patients might respond to different treatments in different ways. Now, when computational biologists explore new data sets, they oftentimes don't operate on the raw data sets because it can be very high dimensional. So for example, with gene expression data, we might have on the order of 20,000 to 25,000 different gene features. And there's often a decent amount of noise in the data set due to various experimental factors. So instead, computational biologists make use of a technique called latent variable modeling. And in these latent variable models, we assume that our observed data, which are the X in this picture over here, were originally generated from some lower dimensional set of latent variables called Z, which we don't observe directly. And in theory, these lower dimensional variables contain a lot of the interesting biological signals and less of the noise that we see in the observed high dimensional data. And so thanks to some great advances from the machine learning community, we can actually learn functions that will take us from these high dimensional X's down to the lower dimensional Z's. And one of the very popular models for doing this is called the variational autoencoder, or the VAE. And so for people who aren't already familiar with VAEs, they look something like this. So we have here our high dimensional observation X. We're going to pass X through an encoder neural network, which then gives us a low dimensional representation of X that we call Z. And then to make sure that this Z actually encodes the interesting patterns that we saw in X originally, we're going to simultaneously train a decoder network that goes from Z and attempts to reconstruct the original X. There's some other details as to what Z exactly looks like and whether you have some priors on what it should be. But this is the general high-level idea of how VAEs operate. Now, unfortunately, if we use a standard VAE with the latent variable model that I showed before, oftentimes we can't capture the interesting variations from those target data sets that we care about. So here I've actually plotted the results of training a VAE on gene expression data from patients with AML before and after they received a stem cell transplant. And so we would expect that there should be some systematic differences in gene expression levels between cells that were pre-transplant and post-transplant. But if we plot the data right here, we see that the representations of the cells are right on top of each other, regardless of treatment status. And so the reason for this is that oftentimes, unfortunately, when we're looking at these target data sets, so for example, like in the cancer scenario, the cancer-specific variations in the data set are relatively small compared to stuff that would also be present in healthy patients. So for example, variations in gene expression data that have to do with demographic factors, like age or sex or race, that at least right now we don't really care about too much. So how can we get around this? How can we actually isolate that cancer-specific stuff? So what we're going to do is we're going to modify the original latent variable model that I showed you before to get what's called a contrastive latent variable model. So now we assume that our data points are generated from two sources of variation. First, we have these Z's that are what we call background sources of variation that we don't really care much about. And we've got a second set of variations that we call S. They're these salient sources of variation. So that would be like our cancer-specific variations that we do care about. And a natural question is then, well, can we learn some function that goes from our observed data X and maps to those salient variations S, and not the background variations that we don't care about. So in order to actually do that, we're going to leverage these background data sets that I've been talking about. So the background data sets, that would be like measurements from healthy people as opposed to the cancer patients. We assume that the data is generated just from the Z's, and that the salient factors S are fixed at some value S prime. And this value S prime represents a lack of those cancer-specific variations. So yeah, just to reiterate, a typical background data set might look like measurements from healthy people, while a target data set from before would be measurements from people with some condition or disease. And there are many kind of natural scenarios in which we collect these target and background data sets. So for example, like in a clinical trial, a background data set could be measurements from people who got a placebo, while a target data set would be measurements from people who got the real treatment. And so using this idea of target and background data sets, we can modify our original VAE to get a contrastive VAE. So instead of having just one encoder network like before, we have two encoder networks, one for our salient and one for our background variables. And then when we reconstruct our data, for target data points, if you follow these kind of confusing sets of lines, you'll find that for target data points, we reconstruct the point using both its salient and background latent representations. On the other hand, for background data points, we only use the background latent variables to reconstruct that data point and replace the salient latent variable values with the value S prime that represents the lack of information. So now when we train our model, and if we embed our target data points into the salient latent space, instead of getting something like this from before, where the pre and post-treatment cells are right on top of each other, we now get a very clear separation between the pre and post-treatment cells, which, given what we know about this treatment, is what we'd expect. And so for this example right here, it was nice because we had labelings for our points, just so we could use this example to kind of validate that the model is doing what we expect it would be doing. We hope in the future to apply this to other data from different biological scenarios where maybe we know less about the underlying biological processes. So maybe like data from Alzheimer's patients where we don't already have labels, but we can use these techniques to discover interesting clusterings or separations of the data that maybe biologists haven't been able to see using more naive methods. So thank you, everyone.
Let's see if we have any questions. Okay, let's thank all speakers.